Imagine holding a human brain in the palm of your hand. Now, imagine that this brain is not a biological organ but a sophisticated microchip. This is the incredible concept behind brain-on-a-chip technology. It combines the intricacies of the human brain with the power of microengineering. Scientists are creating miniaturized living models of the brain. These models are transforming how we study the brain and develop new treatments. Brain-on-a-chip technology offers a powerful tool to unravel these complexities. It has the potential to revolutionize medicine and technology. The brain on a chip is not just a technological marvel, it's a window into the universe within our skulls. Imagine tiny rivers flowing through a miniature landscape. This is the world of microfluidics, a crucial component of the brain on a chip. Microfluidics involves manipulating minuscule amounts of liquids within channels thinner than a human hair. These channels act like the circulatory system of the chip. They deliver nutrients and remove waste products, mimicking the environment of the brain. These microfluidic networks are engineered with incredible precision. They allow scientists to precisely control the flow of fluids, creating dynamic environments for the brain cells. The ability to replicate the brain's fluidic environment is crucial for the survival and function of the cultured neurons. Just like the brain needs a constant supply of oxygen and nutrients, the cells on the chip rely on microfluidics for their well-being. This technology ensures that the brain cells remain healthy. It allows them to communicate and form networks. Microfluidics is the unsung hero of the brain on a chip, providing the lifeblood for this miniature organ. Creating a brain on a chip begins with the building blocks of the brain itself, neurons. Neural cell culturing involves growing neurons in a controlled environment outside the body. These cells are the workhorses of the brain, transmitting information through electrical and chemical signals. Scientists carefully extract neurons from living organisms or create them from stem cells. These cells are then placed on the microfluidic chip. Here they are nurtured in a special nutrient-rich solution designed to mimic the brain's environment. Over time, these individual neurons begin to reach out. They form connections with each other, creating a network of communication. This process, known as synaptogenesis, is the foundation of learning and memory in the brain. Watching these neural networks form on a chip is like witnessing the birth of thought itself. It's a testament to the remarkable ability of life to adapt and organize even outside its natural environment. Imagine a bridge connecting the electrical world of our devices with the intricate language of our brains. This is the role of bioelectronic interfaces in the brain on a chip technology. These interfaces act as translators. They convert the electrical signals of neurons into digital information that computers can understand. These interfaces typically involve arrays of tiny electrodes. These electrodes are placed in close proximity to the neurons on the chip. As the neurons fire, these electrodes detect the subtle changes in electrical activity. This information is then amplified and transmitted to a computer for analysis. This two-way communication is groundbreaking. It allows scientists to not only listen to the activity of neurons, but also to stimulate them with incredible precision. Bioelectronic interfaces are breaking down the barriers between biology and technology, paving the way for direct communication with the brain. Section 5. Listening to the whispers of neurons' electrophysiological recording. Imagine listening to the whispers of individual brain cells. This is the power of electrophysiological recording, a technique used to study the electrical activity of neurons on the brain on a chip. As neurons communicate, they generate tiny electrical impulses. These impulses can be detected and recorded using specialized equipment. In the brain on a chip, electrodes embedded within the microfluidic channels act as listening posts. They capture the faint electrical whispers of individual neurons. These recordings provide invaluable insights into how neurons communicate and how networks of neurons function. Scientists can study how these patterns change in response to drugs, toxins, or electrical stimulation. This allows them to understand how the brain processes information and responds to its environment. Electrophysiological recording is like eavesdropping on the conversations of neurons, revealing the secrets of brain function in unprecedented detail. Section 6.3, Neural Tissue Engineering, Building a Brain Beyond Flatland. The human brain is far from flat. It's a complex, three-dimensional organ with intricate folds and layers. To truly mimic the brain's structure, scientists are turning to 3D neural tissue engineering. This field focuses on creating three-dimensional structures that resemble the architecture of the brain. Using techniques like 3D printing and bioscaffolds, researchers are creating miniature environments that allow neurons to grow and connect in all three dimensions. These 3D models provide a more realistic representation of the brain's structure and function. This advancement is crucial because the spatial organization of neurons significantly impacts their function. It influences how they connect, communicate, and ultimately how the brain processes information. 3D neural tissue engineering is adding another layer of complexity to the brain on a chip, bringing us closer to replicating the intricate architecture of the human brain. Section 7. Controlling the light switch of neurons, Optogenetics and Chemogenetics. Imagine turning specific neurons on or off with a switch. This is the potential of optogenetics and chemogenetics. These techniques give scientists control over neuronal activity. Optogenetics involves modifying neurons to be light-sensitive. Shining light activates or inhibits these neurons. Chemogenetics uses receptors that respond to drugs. Adding the drug activates or silences neurons. These techniques revolutionize neuroscience research. They help study neurons and circuits in behaviors and diseases. Section 8. AI and machine learning, deciphering the brain's language. The human brain generates a vast amount of data. Every thought, every sensation, every action is represented by complex patterns of neuronal activity. Deciphering this complex language is where artificial intelligence, or AI, and machine learning come into play. AI algorithms can analyze vast data sets of neuronal activity, identifying patterns and correlations that would be impossible for humans to detect. Machine learning algorithms can learn from these data sets, improving their ability to predict future neuronal activity and understand how the brain functions. By integrating AI and machine learning into the brain on a chip platform, researchers can automate data analysis, identify subtle changes in neuronal activity, and gain deeper insights into brain function. This synergy between biology and computer science is accelerating the pace of discovery in neuroscience. AI and machine learning are like powerful decoding tools, helping us understand the complex language of the brain and unlock its secrets. Section 9. CRISPR and gene editing, recreating diseases on a chip. Gene editing technologies like CRISPR have revolutionized our ability to manipulate DNA. In the context of brain on a chip, CRISPR allows researchers to introduce specific genetic mutations into neurons, effectively recreating the genetic basis of neurological diseases on a chip. This breakthrough has profound implications for understanding and treating neurological disorders. Scientists can now study the cellular and molecular mechanisms of diseases like Alzheimer's, Parkinson's, and epilepsy in a controlled environment. By comparing healthy and disease-affected brain-on-a-chip models, researchers can identify potential drug targets, screen for new therapies, and personalize treatments based on an individual's genetic makeup. CRISPR and gene editing technologies are transforming the brain on a chip into a powerful tool for disease modeling, drug discovery, and personalized medicine. Section 10. Wireless connectivity, a world without wires. Imagine a future where brain-on-a-chip devices can communicate wirelessly, transmitting data and receiving instructions without the need for cumbersome cables. This vision is becoming a reality, thanks to advances in wireless communication technology. Researchers are integrating tiny antennas and wireless communication modules into the brain on a chip platform. This wireless connectivity allows for real-time monitoring of neuronal activity, remote control of experiments, and even the development of implantable devices for therapeutic applications. Wireless connectivity eliminates the limitations of physical connections, opening up exciting possibilities for using brain-on-a-chip technology in clinical settings, drug development, and even brain-computer interfaces. This seamless integration of biology and wireless technology is pushing the boundaries of what's possible, bringing us closer to a future where brain-on-a-chip devices can be used to monitor, treat, and even augment human cognition.